hello students. So, good morning everybody. So, today we will just go for lecture number 53, considering phosphorus as your non-metallic species in your biological world. So, how we can see the different phosphorus and the phosphorus related molecules, particularly the anions, because we are talking much about these anions. So, what is the most important thing is that how chemistry can control the corresponding character or the reactivity of the phosphorus or the phosphorus based anions, then how the nature can take care of all these things through two different parts. That means, one is the biological part and another is the geological part. So, together we can have the biogeochemistry of phosphorus. So, it also related to the corresponding point number 3 is the phosphorus cycle. So, we have, when we have the phosphorus, we are not talking in terms of the elemental phosphorus, that means the element wise we know the red phosphorus, white phosphorus or the black phosphorus. Is not that, the phosphorus related species which can give you some anions, and those anions how they are important for our biological world. So, most important thing when we talk about phosphorus or the phosphate anions, we all know that the ATP molecules is our energy currency. So, how much ATP we synthesize daily and how much we consume and why this is so important. And also, if you consider that you have the phosphate anion, and we all know in our body also including water, many protonating species are available like the free amino acids and all these. So, if you are going to protonate the phosphate anion, you will be ending up with the phosphoric acid H3PO4. So, it is not that we are producing the free phosphoric acid in our body or our fluid medium, but the corresponding anions are there and the protons are also available. So, we can have some medium which is very much buffering in nature. Then finally, the biomineralization process which is also dependent on phosphorus. So, what are the phosphorus related material we can have for our bone, for our teeth? We know apatite is the name of that particular mineral. So, when we talk about the phosphorus in biological world, we always ask that why nature is considering these phosphates, because this phosphate is very much important for our backbone, for this ATP preparation and all these things may not be so important in handling your dioxygen molecule, but some other enzymes are also dependent on all these things. So, when we talk about the non-metallic elements in the biological world, so we think about the phosphorus after carbon, oxygen, hydrogen and nitrogen. When we talk about that, whether it is involving very frequently in the redox chemistry, that means electron transfer reactions is not. So, it is stable in the highest oxidation state, that means +5 oxidation state. But what we know from our school days, the elemental form, just now I told you the phosphorus as the element, the tetramer, the P4 phosphorus element. So, elemental form usually in the solid state. So, geologist will come and say, oh, I know it from the mineral or the ore, I can take out some phosphate rocks. So, those phosphate rocks, when we treat those phosphate rocks, the phosphate rocks can give you the elemental phosphorus, but that is all in the solid state. So, in the standard temperature and pressure condition, what is available on the earth's crust or earth's surface, this phosphorus is present as in the solid state. So, when this inorganic phosphorus, we can go transforming it to the phosphate anion, and when we transfer it or transform it, that particular phosphate is required by all forms of life. So, it is very important. So, as we considered earlier from our very beginning of our class, the average human body, if it is 70 kg, how much phosphorus you can have in your body. So, adult human basically contains have about 2 pounds of phosphorus, that is about 0.8 kilogram, that means 800 grams of phosphorus in your body. So, that phosphorus is your total phosphorus, whether you are that phosphorus is present in your teeth or that phosphorus present in your bone or that is in the corresponding ATP molecules. But how we can correlate all these basically? The elemental phosphorus we do not have, but we have the another mineralized form which we consider as the biomineral. So, biomineral or biogenic mineral, what we can have, starting from there where we can think about the corresponding species or the compounds which are important for our genetic information transfer, that is your DNA and RNA molecules. So, how the DNA and RNA molecules are also linked to the phosphorus or the phosphate anion. So, we have the living cells, they are utilizing phosphate to transport the cellular energy with the ATP molecules. So, we know that the phosphorylation reaction as well as dephosphorylation reaction is always taking place on the basic backbone of the adenosine triphosphate. So, what you have? You have the nucleobase, you have the sugar unit, then the phosphate unit, and then if the phosphate unit can form the esters or diesters, we get the corresponding DNA and the RNA molecules. So, all these are linked, but what we are trying to talk about today is that how you can think of only the phosphate. Your eyes are now centered on the phosphorus of the phosphate anion. Like always I say that a biological inorganic chemistry is basically devoted to many metal ions, but when we start our discussion about the non-metal ions like carbon, hydrogen and oxygen, so like that of your phosphorus. So, you should be able to tell that how the phosphorus is also important for your bioinorganic chemistry. So, the cellular processes and also it gives the energy due to that of your dephosphorylation reaction and the production of many number of PIs. PIs are capital P, small I is your inorganic phosphates. So, out of all these 85% is deposited in bones and teeth as calcium phosphate in the form of crystalline hydroxyapatite. We know the powder form, if you have the calcium phosphate and if that is present in the powder form, that is no so strong. But if you are able to crystallize it, not only the pure calcium phosphate crystallization, but you can have the hydroxy derived one or the fluoride derived one, the fluoroapatite and the hydroxyapatite, but along with some organic matrix, it can go for increasing its strength. So, in terms of the corresponding phosphorus concentration as well as the corresponding concentration of the inorganic phosphates what we can have in your blood or serum. So, all are present in the soft tissues and in the serum also, but that concentration is not very much, is only 1.1 to 1.4 millimolar concentration. So, we have started our discussion about how much phosphorus you can have, how much can be deposited, 85% can be deposited as your bone and teeth formation. Then the remainder is not crystallized, it is still in the solution, because we get everything crystallized from the solution. When you have the saturated solution, you go for the crystallization. So, what is the remaining or the leftover solution concentration? It is not even in the molar concentration or something, but it is not even less, that means not nanomolar, but it is the millimolar concentration is pretty high. So, pretty high concentration of these things can manage all the biological reactions, because always we know that the dependence of any particular chemical reaction is there on the concentration of the species available to you. So, in the biological system what is available as the phosphate ion, and then slowly from your biogeochemical part, if we think about that geochemistry is talking about on the soil and minerals and the rocks, then how the plant and the other animals can take up all these things, particularly those herbivores which are taking the plants as their food material. So, if plant gets that phosphate, then those animals can also be taking that phosphates. So, the phosphate cycle or the phosphorus cycle can continue in this manner. So, the properties what we can have with this particular phosphate, which are essential for many biological functions. So, I am giving you the statements, read the statement nicely, such that you can have some good information about DNA, RNA and how these ATP molecules and all these. So, these are the basic understanding only why we should read phosphorus. If somebody says that okay, we can go for the phosphorus chemical biology, what you are studying is phosphorus chemical biology, you will be thinking no, it is very complicated one, is nothing complicated. You want to have the knowledge about the corresponding phosphorus bearing biomolecules only. So, DNA information retention, where you retain the information from one generation to the other, you require a linkage which is phosphodiester linkage. So, you should know the constituent, you should know the chemical composition of all these molecules or the biomolecules, then the corresponding structure. So, they all must have the phosphorus, then we know that the phosphate can come, but it is a diester thing. So, if you have the phosphodiester, it is the DNA or the RNA molecule and rapidly repairable when required. That means, ATP, ADP, AMP and the phosphate transfer reactions and the phosphorylation and dephosphorylation reactions are going on all together. So, if we consider that one theory is there that our life started from DNA. So, if we consider that is also true and if you have the DNA world, then the same phosphodiester is again available and which is your covalent linkage stitching together the strands. So, this linkage is very important. So, then you have finally the corresponding hydrogen bonding interactions from the nucleobases, but before that you have the backbone with that of your phosphate diester or phosphodiester linkage. Finally, we will see also the phospholipids. What are phospholipids? We know lipids, but what are phospholipids, whether you have a diester bond or not, and then you can have how the corresponding assembly of those lipids can give you the corresponding biological membrane. So, the calcium and calcium derived phosphate. So, whenever you have the phosphate, we all know our body cannot restrict that okay, calcium should not reach the phosphate. So, we have the corresponding approach of the calcium ions which are cations and the phosphate having 3 negative charges on the phosphate anion. So, it is highly charged. So, if they are coming together, the chances are there they can combine together and form the typical inorganic compound calcium phosphate, and that typical inorganic calcium phosphate compound will have very low solubility. So, when you have a corresponding concentration of these phosphates, free phosphate anion in millimolar range 1.1 to 1.4 range, you can have also certain amount of calcium concentration in our body fluid. So, they will come together, combine and try to crystallize from the medium. So, you can have the corresponding crystal growth for the bones as well as on the for the teeth. Then for the hydrolysis reactions, ATP to ADP conversion or ADP to AMP conversion, we know we will be able to release some free energy for that particular dephosphorylation reaction and we get the energy for that particular purpose. Then we have the signaling. So, the information flow as well as signaling is also dependent on the phosphate chemistry. So, within this particular case also 2 parallel signaling regimes or the areas we can have. So, when we have certain amount of phosphorus available for not only your DNA and RNA molecules, but you think of only say ATP molecules. So, when we have this much phosphorus in your body, you consider your body as a storehouse of your phosphorus based biomineral, it can be your teeth, it can be your bones. Then you think about how much ATP you can have in your body. So, the rule of thumb is very simple, you will be able to remember the way I also remember the average human body is we consider as 70 kg or you can consider it as a 75 kg, but you have only 75 grams of ATP, but it is cycling very fast. So, it is very basically a catalytic molecule ATP to ADP to AMP and is cycling back and forth. So, 1000 times of that amount which is 75 kg of this. So, throughout the day we will be able to make, that means you as and when required or when we need it we prepare. When you take the food material you have the glucose, you have only the store of 75 gram of ATP, but that will cycle for 1000 times. So, if the 1000 times cycling is available, you will be able to produce an amount of total ATP which we require for our day long survival is about 75 kg again. So, it is easy to remember also. So, if you have the phosphorus and the phosphorus linkage, so you will have the POP linkage, that means diphosphate unit. How the diphosphate unit is formed? Definitely we have the anhydride PO of one and another PO will come making the POP bond which is your anhydride bond, is also for the triphosphate because ATP is a triphosphate molecule on the side chain basically. So, when you have the side chain you can have 3 p, 1 p close to that some part and the terminal p's. So, you will have p alpha, p beta and p gamma, and which can have the corresponding thermodynamically favored attack by cellular nucleophiles. So, nucleophiles will come, attack these links basically the chain, the 3-membered chain, and then can go for some very useful reactions. So, not only the importance of the phosphorus in the biology, but several phosphoproteins also can take part in biological activities. What are phosphoproteins? So, phosphoproteins are not so complicated thing. Try to understand that whenever you have some OH function of alcohol or the alkoxide ion, you know that is a typical nucleophile. It can attack the triphosphate or diphosphate to take up the phosphate group attached to that particular oxygen of your nucleophile. So, the phosphorylation of the amino acids can also be possible to have. So, if you have the corresponding phosphorylation of the amino acid residues having some alcohol function, say the serine, the OH function is there. So, that particular part is getting phosphorylated, but you have the free amine function and the free carboxy function. So, the free amine function as well as the free carboxy function what you can have can give rise to the corresponding dipeptide, tripeptide and polypeptide and ultimately to protein formation. But you have the corresponding pendant phosphate group. So, those are basically your phosphoproteins. So, there also we require the knowledge of phosphate chemistry for the understanding of the different metabolites which can be of low molecular weight or can be high molecular weight. So, basically when we have the Pi, capital P and small I, what we write all the time, which is your inorganic phosphate. So, that inorganic phosphate what you can have we consider as Pi, we write in the biological world or the people who are, because it is the abbreviated form, very quickly you can understand is your inorganic phosphate. So, it is playing many roles. It is poorly soluble as calcium sulfate, as just now I told you that you can think about the corresponding calcium salt of the calcium phosphate. So, what we find there? We find Ksp, the solubility product of the individual ion concentration, that means the individual ion concentration of or ionic product we call something. So, ionic product of the calcium concentration and ionic product of the phosphate concentration. So, if your phosphate concentration is pretty high, which is in the range of 1.1 to 1.4 millimolar, you will be able to get or reach this particular value of ionic product very quickly, such that you can have the separation of hydroxyapatite for your bone and teeth enamel formation. So, the central phosphorus atom in the phosphate is basically in the +5 oxidation state and is electron deficient. So, that is why it will attract many nucleophiles. So, higher pH value basically if you have, that means if you have the calcium phosphate and you have a higher pH value which redissolves the phosphate dianion and is the basis for the dynamic bone resorption, basically resorption. So, that dissolution, so it is a continuous process, because the bone we have the corresponding deposition we know, the growth of the bone we know is a dynamic process, but after some time is not the shrinking, but it can have the corresponding bone density, basically we call the loss of calcium. So, it can have the corresponding leaching, because it is always there like a mineral you have and you put inside a corresponding acidic environment. So, in the acidic environment which is less than only pH 7, so you can have the dissolution of the basic anions. So, calcium can be leached from that particular solid material. So, again the Pi can have thus a physiological role, and neutral pH values is a mix of monoanion and dianion. So, what we can have if you have a neutral close to 7 pH value, always we try to consider that our blood pH is only from 6.8 to 7.2, something like that, is not our stomach pH which is close to 2 to 2.8. So, what we can have, you see the phosphoric acid which is the fully protonated form we can have, which is H3PO4, but we can have the trianion at the end which is your PO4 3 minus, and you can have the involvement of 3 pK values, particularly the pK 7.2 which is the pK 2 and the pK 3 which is 12.7, these two are important. So, in many times we can see that you can have the corresponding oxo anions, which are your biological nucleophile, and these corresponding oxo anions can be protonated depending upon its available environment, where you can satisfy the corresponding pK value for dissociation of the proton. So, if your pK or the pH value of that particular medium is less than that of your 12, what you can think, you will, instead of getting the typical PO4 3 minus, it will be protonated in the single step. Similarly, in the second step also you can have the double protonation of the phosphate anion. There then again we know that the enzymes, the phosphorylase enzymes are there, and those phosphorylase enzymes can transfer many electrophilic fragments of substrate to inorganic phosphate. So, the work or the role of these enzymes, what these enzymes are doing basically, they basically control the reaction between the electrophilic fragment to the substrate to the inorganic phosphates. That means, it will attack either the oxygen center or the phosphorus center. Phosphorus center is having a charge of 5 plus. So, it will have a demand for electron density, but oxygen having the charged one O minus. So, it will have the demand for the corresponding positive charge. So, that is why within the system you can have a electrophilic site as well as a nucleophilic site. So, we can have many such reactions which can be controlled by different enzymes, which can also be metalloenzyme. So, one good thing we have studied from our school days, again the glucose oxidation or the assimilation of the glucose for our energy production, where we produce large number of ATP molecules. But while doing so, we know that we can have the glucose alpha 1 phosphate. What does it mean basically? That means, you have the glucose, and we all know the glucose can have some groups, can have aldehyde function, can have the alcohol function, CH2OH function. So, if we are able to phosphorylate that alcohol function nicely, we get the phosphorylated glucose, that is your nothing but your glucose alpha 1 phosphate. So, we can go for the phosphorylase reaction, and that phosphorylase reaction is basically controlled by this enzyme. So, this is your phosphorylase enzyme. So, the mobilization of the glucosyl unit, that means you phosphate is there. Now, you think of in a different manner, that you have the glucose unit attached to that phosphate. So, think of that movement of that glucose unit. So, whether we can take out that glucose unit from the starch or from the glycogen, we know that in our body the unused glucose which we are not burning immediately for our future use or future demand, it can be stored as the glycogen. So, the glycogen is stored, which we all know as the polysaccharide, because many times we take the disaccharide, the sucrose, fructose, all these are disaccharides, but also we take polysaccharides, but we cannot consume many amount of the starch. So, these polysaccharides you have to break. So, the hydrolysis is taking place, and finally, you get the glucose, and that glucose can be phosphorylated. So, all these energy transfer reactions are dependent on the transfer of the phosphates and sometimes the inorganic phosphates. Then PPI, the inorganic pyrophosphate, that means one more phosphate, POP bond is there, at neutral pH exists in large part as tetraanion. So, if you have two phosphate units attached to it, so altogether you can have many oxygens. So, those oxygens can be charged. So, you can have the tetraanion at a neutral pH value. So, whether you are removing that particular diphosphate unit or putting the diphosphate units in some other molecule is important. Then kinetically, the phosphoric anhydride linkage formation is stable enough for pyrophosphate tetraanion formation, that is your all the 4 oxygen centers are charged, that means those oxygen centers are charged to be diffusible metabolite in all cells. So, since it is charged, you have more charge, that means you have more solubility. More solubility means that you can have the metabolite. So, you transfer those phosphates. My main intention at this point may be we want to move or we want to transfer those phosphates as the corresponding solubilized form in different metabolites. So, metabolites what we gain. So, metabolites are important for the burning of the glucose molecules for get the energy. So, if you have this particular anhydride side chain, that means POP bond is there, which enzyme can control for the formation of ATP, NTP, DNTP, phosphate side chain cleavage reaction. So, you have triphosphate, then diphosphate, then monophosphate. So, attack of an electron rich nucleophile at the outside P alpha of ATP constituent. So, that means the terminal phosphorus we can label it as the P alpha, then you can have the P beta, and then you can have the P gamma. So, you see now that if you have the P alpha, so this is your P alpha. So, when you attack the P alpha, we consider it as a phosphoryl transfer reaction, because your Rx, which is your corresponding nucleophile, which is coming and attacking your P alpha, giving the corresponding phosphorylated nucleophile. So, you can have the alcohol function, you can have the glucose function also, and this is the most simple reaction what we can think of for the corresponding phosphorylation reaction. And we consider that is the phosphoryl transfer reaction, which is also true for the typical ATP, giving you ADP and the energy. So, already we know that we require the corresponding coordination of the Mg2 plus, magnesium is binding, then only you can have. So, magnesium is the still bound form. So, magnesium ADP is there, and you basically cleaving the corresponding phosphate group from there. So, we are releasing some amount of energy. Then the other two, that means if you go for the beta as well as the gamma phosphorus center, one will be considered as the pyrophosphoryl transfer. So, 2 unit of the phosphate can be transferred, and the other one is nucleotidyl transfer involve the attack on these two phosphorus centers. So, definitely it will be related to your route for ATP biosynthesis, and that ATP biosynthesis can have 3 common routes, and which can make many times we like your corresponding production of your ATP. As I told you that you can have the throughout the day the 75 kg of ATP we are using with the 75 gram of ATP. So, the membrane embedded ATP you can have. So, altogether 94 and 95% of those amount of that amount of 75 kg of daily ATP requirement under fully oxygenated condition can have the proton transfer potential, protons running down from the electrochemical potential. ATP syntheses are there for the synthesis of the ATP molecules, but for the breakdown which is required for your energy production, which you get in your mitochondria. So, it basically requires the access for the pool of ADP molecules, inorganic phosphate in the mitochondria, such that ATP synthesis can work, and the corresponding net dehydration coupling to make a P beta O P gamma anhydride bond in the ATP. So, ADP or AMP if you have, you bring the phosphate. So, you have the gamma, then you put the beta, and then finally, the alpha phosphorus center. Then the phosphomonoester, what are they? So, bring alcohol as the nucleophile on your for the reaction, the first type of reaction what we consider as the phosphoryl transfer reaction, and the group on the alcohol functionality that gives rise to your glucose 6 phosphate formation. Over 100 low molecular weight kinase enzymes are involved and 500 protein kinases. So, it is a huge number, do not worry about this number. So, we are only trying to understand the basic reactions what you can have based on the phosphorus. You can have the hydrolysis or you can have the formation of the new POP bond. So, alcohols will come and the many other charged species can come and bind to all these cases. So, you can have the different low molecular weight as well as high molecular weight of these phosphoproteins, and the phosphatases will also be there, which will be required for your hydrolysis. Then phosphodiester, the POP, and is required for your nucleotidyl transfers or nucleotidal transfers. So, it can have 2 alcohol groups which are bridging phosphoryl groups, and those bridging phosphoryl groups are basically like this now. So, now instead of attacking the first one, you are attacking the third one. Now, you see the control of the magnesium ion is different. So, you have the adenylated nucleophile. So, if you have any alcohol part or anything, you can put one group to that particular group. So, these are very important reactions for these phosphodiester formation, and the 2 key biology classes of phosphodiester are RNA and DNA molecule, because you have the phosphate and from 2 ends you have the corresponding ester formation. So, in other cases also the nucleotidyl transfer can be useful for your phospholipid formation. So, you have the backbone then you bring this particular part. So, this is your lipid part, and then you bring something else which is your ethanolamine phosphate part. So, ethanolamine phosphate part, if you bring and attach to that particular the central glycerol OH group or O minus group, you get the phosphatidyl ethanolamine, or you can bring the choline also, the phosphorylated choline part, to get that particular phospholipids. So, membrane phospholipids are therefore very important thing also for the phosphorylation reaction and the dephosphorylation reaction, because you can have the polar head groups to the hydrophobic diacyl glycerol backbone. So, this is one acyl backbone and this is the second acyl backbone, and then you can have the phosphate backbone on the other side. So, we have reached to the end where we have seen that the chemistry of not phosphorus, what we have learned from our school days again, inorganic phosphates, its anhydrous monoesters and diesters with alcohol groups of metabolites, because we know the glucose, and if you burn it further you can have the many alcohol, the pyruvic acids and all these things. So, all metabolites are we have. Low molecular weight as the macromolecules, and they dominate and determine the myriad roles of phosphates in biology. So, all these chemistry basically, all this chemistry of phosphorylation and dephosphorylation reaction can give us many good informations related to our phosphate in biology world. So, we basically can go typically for the biochemistry of phosphates within the page of phosphate or phosphorus, you can have in the Wikipedia page, but you look at the biochemistry part of the phosphates and how, what are the biochemicals or macromolecules or biomolecules involving phosphorus, then the book of Crichton. Okay. Then thank you very much for your kind attention.